Beneath this very room, there may be a deadly bacteria that can kill a human being within 48 hours. We live with it, it's in the soil around us and in groundwater. It can enter our body through cuts in our skin or inhaled through our lungs. It can cause skin sores, infect virtually any organ in your body, and if it reaches your brain, is usually fatal. I'm talking about the human disease melioidosis. 1 in 10 people with melioidosis will die, and those that survive may be left with lifelong illness. In the last 3 years, there has been 252 cases of melioidosis in the Northern Territory. This disease is difficult to diagnose, and currently there is no vaccine. This bacterium is now classed at the top of the list of deadly organisms outlined by the Centers for Disease Control. This means it's in the same class as anthrax, smallpox, and plague. It is feared that this bacteria may be used as a biological weapon. From a global perspective, and within Australia, it's important to understand how this bacteria lives in the environment and spreads over time. These areas are still poorly understood. My PhD involves studying the DNA, the genetic makeup of this bacterium. So far, we've collected 7,000 strains from soil and water and from within infected patients around Australia. The process of studying the DNA of this bacteria is analogous to piecing together a puzzle, except in each bacterial strain I study, I have to put together a puzzle made of 7 million pieces. This is a huge task and requires the use of supercomputers to put together the puzzle pieces in the correct order. Once I have a complete puzzle set from each bacterial strain, I can compare them. Specifically, I can look at the difference between environmental and human strains and those from different geographical areas. So far, this research has shown that any strain in the environment has the ability to infect humans. This work could also highlight potential puzzle pieces that could be a breakthrough for vaccines or diagnostic tools. Likewise, could we use these puzzle pieces to catch those responsible in the event of a biological attack? We aren't there yet. In other bacteria such as anthrax, this task is more simple because it has relatively fewer puzzle piece combinations. Unfortunately for us, the melioidosis bacteria loves to swap around its puzzle pieces, leading to many combinations. This work is at the forefront of genetic study around the world. So why am I doing this research? From a global perspective, to find those responsible in the event of a biological attack. But closer to home, in the Northern Territory of Australia, we want better patient outcomes by potentially improving diagnosis and treatment. Thank you.